Hello dear all beautiful students, welcome to very first morning here with the biology first paper class, and today we will go through the solution of biology first paper assignment 1. And already I think you got the assignment. I translated it in your section yesterday, and this assignment is look like very easy assignment because we already completed chapter 1, what the asking for in the assignment, but the question in my hand, I will go through that one, but writing will be maybe the taking the more lots of pace with having, because the very big topics is actually including in there, but basically which way in short format or how can we solve the assignment to get a good figure. Very first time you always follow the evaluation in extraction. How do evaluation on there is 1 2 3 4, in which way, what to maintain in there to get the 4 out of 4 or 3 out of 3 or 2 out of 2, if that question marking. That means you will follow the evaluation rubrics in there. In the right side you will see right side 1 2 3 4 likely to have the assignment rubrics, that 3 category. First, the instruction is given in there. There is DNA to RNA, RNA to protein. Actually the assignment is on the basis of their DNA to RNA, RNA to protein. This one is the assignment formation, very first one. And what to follow the instruction here is mentioning in there. Very first one, there is instructed here the structure of DNA and its physical structure actually mentioning there. And then after the second point, to DNA to RNA, which way to produce or created in that one, which way. We know already that is the transcription process actually, transcription, and we will go through detail about the transcription, though I studied and I make you understand in before when we completed the chapter 1, the detailed study on there. And the RNA to protein, it is the translation. We know about that translation. Okay, so actually there are the 3 questions to solve accordingly properly, but 3 questions. First one is the physical structure of the DNA, to draw that one and with marking different parts, and the process of producing or creating RNA from DNA, transcription process in details. And third one is RNA to protein, that means the translation process is going on. On that time we will go through the 3 solving question, first, second and third, with drawing figure and explanation. Yes, so you can think about, okay, I will draw the figure in generally and I will go through getting the good marks. If you think about likely to have that one, I think I tell you, you will not following up or follow up that one directly. Just I ensure it here for you and follow me to draw. And that one, which one? At first we will write about, at first, very first one assignment start when it will be on there. So you will give some idea, because their learning outcomes, there is a learning outcomes is mentioning in there. We can understand about the structure of DNA and RNA, mentioning RNA, but when instruction is given, they are asking only to draw the figure of physical structure of DNA, but starting idea is on there. Okay, so very first one, you will tell something about DNA and RNA, then structure of DNA. That means DNA, we know about that it is a deoxyribonucleic acid, deoxyribonucleic acid, and it is the chemical chemical basis of heredity. Almost all living things, especially for animals. That means in case of animals, especially for animals, the heredity materials and chemical basis of heredity, DNA is present on there. Just simple writing on there, very simple writing. Since the Facebook live video in there and Zoom, I set the two device for making sure to complete that one. Okay, and why we need to write something about RNA? Why it is needed to write something about RNA? Why we will write about RNA? Because in the starting point, in the learning outcomes, there is asking we can know about DNA and RNA. Yes, of course, no need to have any panic. Ribonucleic acid, very short way, because in the 40 minutes, how can I complete in total writings? But gist and keyword I will mention in there, and you can explain up in the question on there. Ribonucleic acid, and it is also termed as heredity materials, but it is also act as heredity materials where there is no DNA. There is no DNA, especially for plants. Yes, our starting part is now okay. Starting this one, but if all of you copy me here, likely what I have done in there, it will be the same writings. So by your own self, you can just starting and adding with that. Starting DNA and RNA, we write something, and now the structure of physical structure of DNA are described or explained in below. Okay, starting that one, and then now we will go from a starting part. After then you will write, because I will draw the figure on there. Yes, it is not have any panic that it will be very hard topics or likely that. Very interesting topics. We can easily solve that problem in accordance with. So physical structure, below you will write on there, physical structure of DNA. Yes. So which figure? There is a two figure already we have done in there. Best way to write the figure. I will draw the figure and try to make you understand what the point to follow up in here. Okay, I erase that one also. I will need to draw the figure in accordance with there. First of all, we will make sure anything, any starting part. Suppose I start from here. This one, that one, by my own self. Okay, fine. Then I start also likely to have. Okay, I make it this way, and that way. Suppose I do it in this way. There is no panic about that. Sir, is it a DNA structure? Actually, it is a DNA structure. You can make it double. Okay, and it is better to have the straight way to draw. That will be more better to have in there, and make it double. Okay, I make it double in there, double layered. Okay, I make it just for having a structure. You can draw with pencil, so you can erase and straight likely to follow on there. Just making sure of the drawing of likely that structure, and it is 3 prime, it is 5 prime, and in between them there is a adenine with the thymine, guanine with the cytosine, again cytosine. It is not mandatory that, sir, ATGC I will give it in there, or not, or likely to have, which way I will give it in there. So it is one. Then suppose it is your own choice that which one you will write first, but keep it in mind, when guanine with cytosine, the triple bond of hydrogen bond in between them, and when thiamine and adenine, double hydrogen bond in there, you can draw up in this way accordingly. Just innovative. Everyone, when they will copy from the book, their drawing style and everything to be same. So just getting some innovative, because assignment condition, first condition of the assignment is actually to have innovation. The student use themselves for by their own self, they use that one or not properly. Okay, so when you will draw by yourself, write something about them, better to draw, better to write accordingly. Okay, there is a structure. This is a structure. So sir, I draw this one only, this one with another figure I already taught you in before. Okay, beside then you will draw suppose adenine and thiamine. Okay, and this one I gave it there this way, and this one to this way. Suppose adenine and thiamine. Okay, in between them the double hydrogen weak bond is connected on there, and here in the in that place, okay, I connect this one, and look at here, oxygen, and then this one I draw, and from that part a phosphate is connecting on there. Pentose sugar, I draw a sugar in there, sugar in this side. Okay, and also having that side, since I take very concise place. Okay, no matter what. Okay, I draw likely that one this way formatted. Okay, so there is numbering is given. It is not mandatory the number. Okay, by the way, you can give it in there, the numbering of there 1 2 3 4 5, or likely that sugar in there 1 2 3 4 5 on there. I just erase that part only for there. Adenine and thiamine, the setup. Sir, can you set another one thing, guanine and cytosine? Look at here, the pentose sugar is look somehow big and small because there is not a space. You can getting the same space, or no need what happened in there. Okay, then after, look at here, from that part, connector is the phosphorus in them, and again I will make sure guanine and cytosine. Guanine this way, and guanine part is likely to have in that way, and triple bond 1 2 3, and connecting with the cytosine, guanine and cytosine in triple bond connection, and both of them having the same structure with having that part. Okay, this one, that means each and every figure will be same. This one, okay, sugar, same way you can make sure in them, and from that part to that part there is a phosphate to connect in there, and from there also having the same figure, this one, there right side when I draw it is look like because of a small space, that one. So phosphorus from there and connecting with this part, phosphorus, after drawing, and in this way same thing you draw once again. If it is that carbon, third carbon with fourth carbon connectors, same way you can draw adenine, thiamine, guanine, cytosine. Follow up, same way will go through there, because we know that a nucleotide, DNA, is nothing but a polynucleotide chain. Nothing but polynucleotide chain. That means nuclear. What is the nucleotide? We already know about that. Nitrogen base, adenine, guanine, cytosine, thymine. Nitrogen base, of course, be there. A pentose sugar and phosphate. Nucleotide equal to nitrogen base, pentose sugar and phosphate. Here we already draw nitrogen base, but basically why I draw this type of figure? Not only two, you will add more of them. Again, once again, adenine, thymine, guanine, cytosine, same way you will draw at least 3 or 4 times. Okay, 3 or 4 times, same way to connecting and chain, making 2 or 3, 4 or likely that. It is okay. Then look at here. Everyone, some student asks me, sir, is it the adenine, thymine, guanine, cytosine or cytosine, guanine, it is a nitrogen base? Yes, it is a nitrogen base, but actually it is a nucleotide, because in between them, in the double helix membrane, when we see this structure, this sequence is in there, not only the nitrogen base, because this is a nucleotide chain, nucleotide chain, nucleotide chain. In this way, the polynucleotide chain, many of the nucleotide chain. So this is a DNA. So that beside then this figure to be drawn also. Okay, very interesting. And when we will go through understanding about that what type of structure or what type of features you will write, you can get help from the book also and some idea and keyword from my class. Okay, now I will write something. I draw the two figure for first question answer. Here I just erase that part for writing something or give you some idea about what will you write in there. Okay, that one. By the way, what will we go through about writing in there? You can see about 5 prime to 3 prime in there, and look at here some point we will mention in accordance with here a complete twist. If I find out a complete twist on there, this one is 34 degree angstrom. Okay, 34 degree angstrom, and then there in between these two 3.4 degree angstrom, 3.4 degree angstrom, or you can in size of nanometer 0.34 nanometer also you can write in there. 3.4 nanometer you can write also in there. Okay, 20 degree angstrom, this is a diameter, 20 degree angstrom, or 2 nanometer you can write in there. Okay, already we have written in accordance with that figure. So of course below figure structure of DNA, we already have written that one. So keyword, I need to go through the 3 of the solution of that question. So structure, what will it is a double normal. Look at here, you can get idea from me. Double helix structure, DNA structure we already got. Doubles, this is one helix, another helix, double helix DNA structure we can got in there. Then this type, the DNA, a complete twist. We know that it is a polynucleotide chain composed of you can write about them, the composed of polynucleotide chain, since the many nucleotide in there. Okay, and in between them we can see a complete twist. A complete twist is almost 34 degree angstrom in there, and many students don't know about what is the complete twist. Look at here, if I cut this part from here and this part from there and bind together, a complete twist. Some students think that this is a complete twist. No, this is not complete twist. This one and this part, when we bind together, that is a complete twist. Complete twist, circle, full circle on there. A complete twist is of 34 degrees. So I write very short way. You can write in likely have elaborate the complete twist of DNA double helix. You can find out the 34 degree angstrom or 3.4 nanometer likely to go on there. Okay, then complete DNA twist you can write already on there, and two nucleotide beside having distance of or length of, you can write about distance of 3.4 degree angstrom. And though many students keep it in mind by memorizing, no, look at here, there is a 10 nucleotide in a complete twist, in a complete twist, in a complete twist, there is 10 nucleotide, 10 nucleotides. Since if it is a complete twist, there is a 1 2 3 4 5 6 7 8 9 10 though I draw only 6, but it is almost having 10 nucleotides, a complete twist having. And in between two nucleotide, this one is a nucleotide if it is there, and this one is the nucleotide. In between them 3.4 degree angstrom, because if the total complete twist is 34 and in between them 10 nucleotide, 34 divided by 10 is 3.4 degree angstrom. We got it in there. Yes, the nitrogen base is connecting. Now you can write the nitrogen base. Adenine connected with thymine, double bond, guanine connected with triple bond on cytosine, and finally you will diameter of two strand is 20 degree angstrom, and direction start, that means the helix direction will be in 3 prime to 5 prime, 3 prime to 5 prime, and finally we get the 5 prime to 3 prime. This is the A number answer easily, and I just tell you in briefly going through in there. A number answer, DNA double helix is going on there. Transcription. Now the second point about to know about the transcription is going through. Which way I will go through about then the transcription. What is the question? I just take the question once again. Just a minute. Okay. There is a process, and transcription process, whatever on there. Second point, the transcription process, which way it is to be gone, or it is to be up on their transcription process. DNA to RNA, DNA to RNA. It is the process of transcription. Transcription. I will just having to draw here in accordance with just a little bit. Okay, I start from here. You can follow me to draw that one better. That one, and I will keep it in there, likely to have. Okay, this way I just draw that one and make it double, make it double layered, and then after another one I will start on there in this way. Follow up, just follow me. I will make it double also, this one to know about there. This way. Just follow me that way. So at first give it that one. I will just make it sure to draw the figure once again. Okay, and then after finally another new RNA strand to get it in there. new RNA strand, new RNA strand I draw here. Just follow me to draw, dear student. Yes, it is a new RNA strand, almost on there. Okay. With having to better drawing, forget it back in there. Okay. And there is a sequence we can give it in there, 3' to 5' starting on there, and 3' to 5' on there, and 5' to okay, new RNA strand. This is the new RNA strand. You can write and marking new RNA strand. Okay, you can remember the many of the step during the transcription process. I going through on there, many of the process on there, but there is only single figure you can draw on there. Okay, this is A T G C. In there you can draw likely that G C G C A T, or likely in there G C or C Z, and here A G C T T A G C. Randomly you can give it by your own sense, and then after here in between them the binding part also A, maybe A T C G G C. Likely your own way you can give it in there, the binding part. Other part is the single helix on there, and here also we can get it the GC A T G C C G T A, likely that. In this way you can draw suppose C G G C T A double bond. This way you can draw the figure, and below the figure you will write in there and this is a template for DNA, new DNA and template. Okay, so figure, only this figure you can draw for transcription process. Only this figure is for figure transcription process. Figure transcription process, and what way we will I already give you some points already and starting points on there, in which way we already tried to understand about the transcription process. The initiation of course will be in there, the follow up the process, initiation, then the second point you will write the elongation, okay, elongation in there, and mRNA strand elongation, actually mRNA strand elongation on there, that the process. And number third it will be the termination, and finally you will describe about the finalization of mRNA strand, finalization of mRNA strand. So only I will write this one, sir? No, you will write the explanation of each of them. So what is the explanation? I will use some keyword for better, and it is better whatever I took the class and I have written in the transcription, visit the transcription process, but there I draw a big figure, 3 or 4 step figure. Skip that figure, you will draw only this figure, and just writing what I write up on there, you will go through on there. But actually there is something to add. I can add in there the 4 processes. In case of the initiation, you can write the starting RNA polymerase enzyme. There will be the RNA polymerase enzyme, RNA polymerase enzyme. Start with the initiation of the transcription RNA polymerase enzyme, and then there also you can write about the promoter also in there. Promoter, okay, promoter in there. Promoter, it is just bind with the RNA polymerase enzyme, and the twisted part, what is the result of it? Twisted part unbind when RNA polymerase enzyme, the promoter where the DNA sequence start on there, when it is binding, then there will be unbinding. Suppose we start transcription start from here, RNA polymerase enzyme, if it is on there, it connect with the promoter, and with the help of this enzyme, the twisted part, look at here, it was the twisted, but here unbind. It is starting on there, initiation on starting part, okay, and there is unbind, and 20 base pair actually unbind instantly. You can write on there 20 base pair, and then after okay, the main promoter actually contain the promoter. What is the promoter? Where it will be unbinding on there. This is called the Tata box. Tata box means thiamine adenine. It is unbinded, but sequence to change after then. Suppose in there is a TA thiamine, the binding part. It is now change when it is unbinding part. We can see on there. We will see the TA. Just for example, TAT, AAT. It can be happen likely that. Look like why, sir? I don't understand this one. Yes, I tell you, make promoter. Suppose there is a promoter, RNA polymerase enzyme, starting transcription here. So with the help of RNA polymerase enzyme, promoter connect with them, twisted part unbind. It is no matter what. If just example, if it is the TA is in between them, TA 2 base pair, we can say in there. So when unbind it will be, that means thiamine adenine, it can be sequence to be changed. So it means adenine and thiamine are not directly as like as the previous, where they are binding together. This is to happen. But if you don't write this one, it is initiation is starting on there. You can write accordingly. Then after RNA, second point, elongation. Look at here, elongation. I just write some keyword. Second one, elongation. What will you write here? Elongation. In case of the elongation, yes, the complementary, there is a strand, new mRNA strand. I will write the keyword. You will better to visit my previous class, what the process I write in details on there. Okay, then mRNA strand, move on forward in there, where template of DNA, template of DNA template. One is the mRNA strand, another is the DNA. Move on forward and following mRNA, and when it is completed in there, mRNA strand elongated, mRNA strand elongated in there, mRNA strand. Look at here, the twisted part now unbinding in initiation very first time. Now it is move on, and mRNA strand is starting in there, but when it is move on, the transcription process go forward. So template means where the mRNA transcribe of that template of DNA, it can move on with the mRNA, so that the mRNA elongate. Time it was in there, but when it moved forward, it is enlarged in size, unbinding enlarged in size, and when it enlarged in size and mRNA move forward, and template of DNA is also moving forward. Likely that you will not only for two, I just give you keyword. With the help of that one, you can write from book more of the points, likely that. And finally, what happened in this case of finalization or termination, you can write termination. Okay, now termination, what say in there? Yes, now RNA polymerase move forward. RNA polymerase move forward. Yes, when it is moving forward. So that time, what happened in there? The termination base sequence. The termination base sequence move with mRNA. Base sequence move with mRNA. Yes, and finally, when it reaches at the ending point, the transcription completed. What I write in the keyword? Yes, in this way. Now this one to binding on there. Now RNA polymerase enzyme move forward and unbind the forwarding twisted part. Same figure will be on there, but the mRNA strand, what we adjoin in there, they will enlarge in size and move forward on there, going through on there. Okay, directly going through on there and moving out to sequence on there. And after then, when it is ending, RNA polymerase, suppose it is the initiation point of transcription, it is the ending point of the transcription. Suppose this one, when it reach on that plane or that places, so all the twisted part. Now once again the following part is binding, and we can collect. It is the time to collect the mRNA, because we need to make them RNA to produce an elongated part to find out from there. So finally the termination, I just write keyword. Once again I tell, don't write only 2 and 3 line. In the book you can write more information added, but basic important thing what is happening in there, I just tell you in there. And finally the last one. In the last case about that, it was in the pre-mRNA to final mRNA. Yes, now when it is elongated in size, so we will counting on there, that splicing, collect the pre-mRNA, final mRNA, what was starting in the pre-RNA, collect the final mRNA, then splicing means cutting and sequence. This mRNA I collected from there, and splicing and coding sequence to set up. Coding sequence. That means I collect this one, but now there is a code and barcoding and having the sequence on there. I will collect that one, and finally finally I get it back on there, and splicing. Splicing means to cutting, making setup. It is starting is not going well, and then coding sequence to maintain, and finally we get the final mRNA, and by the process of transcription. Short way I just preview here. I think you understood. Now we will try to understand about mRNA to translation process. What is going on there in case of the translation process? In the short way, I will make you understand. Just very short way. This is a transcription, and then what is the translation process? Number C. This is the C, means the translation process. Translation process. Translation process. And look at here. We can write accordingly in there, in which way to going through in the translation process. Yes, you can draw a figure likely to have in there. Okay. There is a format EPA. Suppose I draw that one for translation process. You will write very first time that by process by which RNA to protein is produced. That means protein is produced from RNA. This process is called the translation process. Starting part you will write on there. Okay, this EPA is connecting with, suppose this one, the EPA. What the EPA I tell you, just wait a bit. Okay. This is the point. Methionine initiator. I tell you, this one is a methionine initiator. You cannot forget about my lecture when you were very first time in the class 11. I told you to visit that class. Already Facebook group is uploaded for better writing. I detail their study because here we cannot going through the much way. Okay, tRNA in there. Then after, what happened in there? Yes, I strand. What is our strand in there? And EPA will be in there. EPA, and I can say here G, suppose AAA. Okay, the mRNA strand. What we get in the transcription process, this one, suppose this one is the mRNA we get, and we transcribe this one. Okay, and then okay, this one for transcribe that one. Yes, the ribosome strand, ribosome strand on there. Very first one, and this one the 5' end, and this one is the 3' end. Okay, then after translation initiation here, what and how I will just tell you. Just a minute. Then we can see in the next figure here. You can draw likely to have with me. Just connecting with them. Yes, and EPA, yes, on there, and the transcribe is already started here. Transcribe already started here, and okay, understand. Just a minute. Okay, just follow to draw me here, like me here. Okay, this one cover, and finally the capturing part is likely that. Yes, amino acid and tRNA. It is the second sequence in there. Amino acid and tRNA is going on there. Amino acid and tRNA is moving on there, and this one is the amino acid. Already we have given on there the G in there, and UAA 5' to 3'. yes, on there, and it is a stop codon on there, yes, and it is the elongation. First one, initiation, then elongation. Okay, initiation and elongation. Second point, it is the initiation. First one is the initiation, and this one is the elongation. Okay, elongation, that means the mRNA strand is elongated in there. And finally, finalization or termination, we can write in there. Finalization or termination. Finalization we can write in there, the third point. So okay, I draw another way, another things. Okay, that one will be likely that EPA, okay, and then same thing, okay, just having to connect with me here, what is happening in there actually. Okay, in there UAA, and just follow me this one, and here polypeptide chain to start in there, likely that I just draw likely. Please follow that one, and getting some space, because I have a short board, I cannot spacious here for making spacious, so make it a space to getting on there. 5 prime to 3 prime we can write in there, and here AA is on there. So it is the number third is finalization or termination, you can write finalization. So what will we see in the initiation, and what is the actually exit? E is the exit peptidyl attachment. Exit peptidyl attachment, it is EPA, exit peptidyl attachment. Why it is mentioning there? The site we need to make sure to mRNA, we can make sure the protein means the unit of the protein is the amino acid, and we know the 20 types of amino acid in here, and having the sequence on there, AUG, UAA, UCG, likely that we know about the part. First part, we need to make sure the exit peptidyl attachment factor here, add with methionine, first amino acid to adjoin in here, EPA, and the strand will going through a sequence on there in the initiation part. So starting initiator tRNA, this is the ribosome strand, since the ribosome will help to synthesis the protein. So here initiation, very first time what we say, initiator tRNA, there is a tRNA is the initiator, methionine initiator. Okay, initiator tRNA, initiator tRNA adjoin methionine with ribosomal segment. Yes, very first time ribosomal segment, and that ribosome is already going through, and this complex is going through. There is a codon there, non-start codon, AUG. When we will get the AUG, the codon, look at here, AUG is the start codon. I just give you once again and repeat, I just give you guideline, taking some information from book and follow that one and draw the figure likely that. tRNA, EPA, set up a methionine initiator with having the amino acid and cover with the ribosome strand, because that ribosome can ensure the protein synthesis from mRNA. We know about that. That ribosome is the main organ to have the protein synthesis, to do that one. So ribosome strand on there. up to when and which way, or long time or after, then when did we didn't get about the AUG, the start codon, up to then they will sequenced once again. First one, the initiation, there is a 3 point or 4 point also be in there. Yes. Then look at here, this part I tell you, tRNA already transcribed, try to transcribe with them with the mRNA, that means elongation part in there. tRNA transcribe with mRNA, transcribe with mRNA up to then the site they are making peptide bond. Look at here, tRNA one, another one is adjoined here. We already got it, suppose a start codon, it is initiation, and now it is the sequence to they make them their number tRNA adjoined with the mRNA EPA site, and increasing in number, and finally what we get in there, they're making peptide bond, because protein sequence, DNA is the polynucleotide, that means many of the nucleotide on there, and peptide bond means peptide bond to form the protein, so that we need to get the protein finalization. In that case we get through that. I just so dear student, it is not have enough time to explain up. I just give you the keyword, please not only write this point. With this you can get idea and having the point with collection from the book on there. And finally when we will get, when this one is moved from here and their tRNA adjoined and transcribe with the mRNA, the coding sequence is also be there, the different codon is also connecting on there, and finally we see that the UAA, when this transcription part get the UAA stop codon, that means a long time it is transcribed, and many of the peptide bond is peptide chain, polypeptide chain is connecting with their tRNA mRNA having the sequence. UAA EPA is adjoining with them to follow up the mRNA, and finally when we get in the mRNA sequence, the UAA sequence, that means the stop codon, that finalization, get the UAA stop codon and complete the transcribe of mRNA sequence. And finally the I just tell you AA, AUG, UAA, UCA, in this way the peptide bond, the lots of amino acid sequence are finding on there, and amino acid, many of the amino acid can form a long protein chain. In this way mRNA can form the protein through the process of translation process. That is all about the assignment, first week assignment of 11 English version. I hope you understood, though it is a very big process. I give you some idea, some keyword, using that keyword information from the book with drawing figure, make it a good assignment and cutting a good figure in the exam. Well wish to you, everyone stay safe, stay sound. That's all about today. Allah Hafiz.